Hello, good afternoon, everyone. Thank you so much for joining, and I had a chance to look at your program, which looks great. I heard that there were some hands-on modules that you're learning as well as these lectures. So what I want to do is give you a little bit of information about the research that my group does, really try to show you how we use these microfluidic and nanofluidic systems and materials to answer questions that are relevant to biology and clinical medicine. So my research group, we do focus a lot on tools design. I actually teach engineering design, the process of engineering design to our undergraduates, and then I also teach graduate courses as well. But what I want to do is use that framework to show you how we pick problems, how we tackle them, some things that have worked, some things that have not worked, and show you this process of iteration and how that's very important in engineering to getting to solutions that actually meet what we call unmet needs. And what I want to do, I had a chance to look at the participant list, which is great. I wonder how many of you here are would not call yourself an engineer. Could you raise your hand? Perfect. And then the rest of you would call yourself an engineer. Fantastic. So I've tried to make this talk be both relevant to engineers and people who would not call themselves engineers as well. What I do hope is that if you have questions or you have thoughts or suggestions, that you'll just raise your hand, and maybe we can do that during the formal presentation as well. Please don't wait till the end in that. So one area that we've been focusing on the past couple of years are these precision analytical tools. So as an engineer, I'm interested in design, so solving problems in a methodical and optimized way. We're also really interested in measurement science, and that's really, I would say, what my passion is from the standpoint of research and engineering, this measurement science. This is a picture of my research group, and again, we're at UC Berkeley. So what I want to do is start with sort of a framework for the type of problem that really motivates us to develop new measurement instrumentation. And this particular problem is actually a pretty big challenge and one that we're making headway on, and I'll use it to frame most of the talk today. So as an engineer, one of the things that we often ask ourselves is not necessarily what is the overarching hypothesis that we want to test, but where are there gaps in capabilities that people may need filled, right? And so one way to frame that in the work that we do is, where are disruptive measurement tools needed? Where could we develop, invent, and implement tools that would change the way people understand biology or medicine through new measurements, right? And so that's a question that I've asked myself many times, and I often pose to my group. And certainly we don't sit in our group meetings and talk about it. We also go out into the field, if you will, right? And for us, that is spending a lot of time in clinical medicine facilities. So we collaborate closely with Department of Surgery and Oncology at Stanford. Also collaborate with global health medicine at UCSF, University of California, San Francisco, these different areas. So we spend a lot of time actually in the field, if you will, trying to understand how people currently do what they do and how they wish they could do things, or in the end, what they wish they could do. And then we figure out how to get them there. So I will argue that over the past decade, or a few decades recently, there have been tremendous measurement advances in the nucleic acid space. One way to frame this is to think about the genomics revolution, right? That really is changing our understanding of biology, so making these genome-wide measurements. It's also starting to change the way we approach medicine and treatment, as well as making prognoses about individuals who are ill. When we look at how those advances have moved forward, one underpinning aspect is new measurement tools, new abilities to make measurements of single nucleic acids across many, many, many different samples and different cell types and questions. And so one area that we feel like is lagging tremendously in innovation in terms of tools for measurements and new ways to make measurements is complementary area, which is protein measurement science, right? So we can understand the genome. We're starting to develop tools to understand the transcriptome. What about the proteome is what we call it, right? So the repertoire of proteins that are expressed dynamically in individuals and in different compartments in different ways as well. So from a clinical medicine perspective, one reason you would be interested in making protein level measurements is that proteins are dynamic, right? They change, and they change very fast in response to environmental and therapeutic factors. In fact, many drugs that are used in the example of cancer actually target proteins. Those are the targets. And if you start looking at proteins from the diagnostic perspective, you might think being able to make real-time measurements of proteins might give you some indication of how an individual is responding to therapy, for example, right? So you can think of proteins as being just a reflection of the organism that is dynamic and is really close to the biology that's happening in the organism at that specific time point. Whereas the genome and the transcriptome are a little bit more static in terms of the information they carry, not quite as dynamic. The issue with proteins are, as I put up here, they are notoriously difficult to measure. So I mentioned this fleeting aspect of the proteins. They change abundance levels, they have modifications, they complex, they decomplex. They're always changing state, if you will, as well as other physicochemical characteristics. There are also many, many more forms of proteins that you could expect than you would for nucleic acids, right? So you have all these different types of post-translational modifications, co-translational modifications, these splice variants, just many, many different forms. So oftentimes what we think of as one protein is actually a whole family of proteins, right? And so that can be difficult as well. An area that's really intrigued me, especially with an eye towards clinical medicine, is this area of isoforms. So proteins that are, again, classified as a single biomolecule, but actually have these various forms that are manifest for different reasons. Now, in the case I'll take here is a very specific example of HER2-positive breast cancer. Who here has heard of HER2-positive breast cancer? Could you raise your hand? Okay, so there's actually several different major classifications of breast cancer, just as there are other cancers as well. So there's ER, estrogen receptor, PR, progesterone receptor, HER2, which is this ERBB2 protein. It goes by both names. And basically, an oncologist, or a pathologist to be more accurate, who looks at a tumor that's been resected from an individual will stain that tumor slice to see which of these proteins are expressed. The ER protein, is it there or not? The PR protein, is it there or not? The HER2 protein, is it there or not? And so using those classifications, the pathologist can communicate with the clinician to say we should use this therapy because we know that HER2-positive breast tumors respond to this therapy well, right? So that really informs the treatment regimen, so the classification of the cancer. So at the biomolecular level, what does that mean? So if we take a look at the cell membrane, so this is outside the cell, the extracellular region. This is the cytoplasm, so inside the cell. This is, for the engineers in the room, a very simple diagram of a protein. I've tried to make it as simple as possible just to call out the important factors for this discussion today. This is known as the full-length HER2 protein. Again, ERBB2 is the alternate name for it. It is a transmembrane protein, so it goes through the cell membrane. In this case, you might think of that membrane as a tumor cell wall. And then a membrane on a tumor cell. You have this extracellular domain, so there's a small region of the protein that hangs out outside of the cell. It's on the surface of the cell. And then you have this tyrosine kinase domain, which is a catalytically active domain that's inside the cell, so in the cytoplasm. This particular form of HER2 we call P185. We call it P185 because it is 185 kilodaltons. So that's the size of the protein, right? And 185 kilodaltons is actually a pretty big protein. I don't know if any of you know of GFP, green fluorescent protein. Have you heard of it? That's a 27 kilodalton, okay? So that's a moderate to smaller size protein. This is a pretty good size protein. Antibodies are about 150 kilodaltons, and those are large molecules. Well, the point we made here, this particular type of breast cancer and other HER2-positive cancers was the first type of cancer where a targeted cancer therapy was developed and approved by the FDA for treatment. So this particular therapy is Herceptin. I don't know if any of you have heard of that therapy before, but it is a drug. It is actually a humanized monoclonal antibody that is used to treat patients with HER2-positive breast cancer. And that's because Herceptin antibody targets this domain. Basically, that antibody recognizes that extracellular domain and latches onto it, blocking that domain, which inhibits some other processes that lead to development of the cancer, and also can be used as a carrier to carry chemical drug treatment to that cell as well. So it can actually poison the cell. It can deliver therapy to poison the cell. The issue is this HER2 positive treatment is not effective if this protein is missing the extracellular domain. So you're noticing that black box is missing here, the extracellular domain. There's no surface receptor for the antibody to latch onto, right, or to bind to. So it turns out that HER2 protein, there's the P185 form with the extracellular domain, and there's actually a whole family of isoforms that lack the extracellular domain, okay? So you see everything from the P110, so it's a little bit smaller, it's missing that region, but it's still transmembrane, and then all the way down to other truncated or shortened forms that don't even have that little transmembrane portion at all. And in fact, some versions of this protein are in the cytosol. Some of them are actually in the nucleus of the cell. So they can be in different regions of the cell as well, but all of them lack this receptor on the surface. So what does this mean? From the standpoint of clinical medicine, that means any breast cancer that is HER2 positive that doesn't have this small surface receptor, it is not going to be treated by Herceptin. This first FDA approved HER2 positive breast cancer therapy is not going to work, right? And so those patients should not be treated with that regimen. The issue is, so it doesn't work against those other isoforms. The issue is, from the perspective of pathology and clinical medicine, we have no way, very limited ways, to make measurements of the presence of these truncated isoforms. So it is difficult to what we call differentially diagnose an individual. Should they be treated with Herceptin, or should they be treated with something else? Okay? So from our perspective, the research that my group does, we're very interested in developing measurement tools that could help a pathologist, for example, be able to say, yes, it's HER2 positive breast cancer, but it doesn't have this domain, so it should be treated with a tyrosine kinase inhibitor instead of Herceptin, right? And so that's the type of problem that we work with. The big issue is pathologists, they use a technique called immunohistochemistry. It's basically a stain. So I mentioned ER and PR and HER2 positive cancer, right? They look at the tissue slice. What do they stain the tissue slice with to be able to make that classification? They stain it with an antibody that contains some sort of tag or label, right? So if that antibody binds to the tissue section, you know that HER2 is there, right? Because you're going to see it under optical microscopy. If it binds to ER, you're going to know it's there because you're going to see that signal light up as well. Well, the whole crux of the issue is pretty much every type of assay that is currently available in contemporary biology and clinical medicine for proteins at the single cell level is an immunoassay. And that means essentially we rely on antibodies for every single one of those measurements we want to make. How many of you here have ever run an immunoassay, immunohistochemistry assay, have ever used an antibody in an ELISA, for example, to make some sort of measurements of proteins? Okay? You know that your measurement for your protein is only as good as the antibody you're using as the probe, right? And some of the big issues with the antibodies are sensitivity, so being able to get enough binding so you can read out low levels of protein, but also something that we call the analytical specificity. If that antibody recognizes off-target proteins and reports a positive signal, you have no idea how much of that signal comes from your target of interest and how much of it is what we call cross-reactivity, right, or off-target signal. So antibodies are really the crux of how good these assays are, these so-called immunoassays. And what I've done here just to help illustrate this is, with my fantastic artistic skills, I have drawn a cell that includes a nucleus, and it has the cytosol, and here's the cell membrane. These circles are different forms of proteins. Again, for engineers in the room. And the Ys here are those antibodies that come in and recognize specific proteins. And what you can see is that in this particular cell, we've stained that cell with an antibody. So we performed an immunoassay. It's green, which indicates it has some sort of fluorescent label on it, let's say. And we see that this particular antibody not only recognizes the P185 transmembrane HER2 protein, that's this black oblong here, it also recognizes the truncated isoforms that are in the cytosol, and it turns out it recognizes another protein that we have no idea what that protein is. It's a cross-reactive signal. It's an off-target signal. So when we do something like IHC, we don't image all the way down to the single antibody level, right? We take a fluorescence image of the cell, and basically what we see is there's a positive signal here, right? The antibody is binding. It's binding to the transmembrane protein, it's binding to the truncated isoforms, and it's binding to some cross-reactive signal. That's not what we want, right? We just want to know signal from the truncated isoforms versus this transmembrane protein. That, I would argue, is one of the biggest challenges in modern analytical science for proteins to date. This being able to identify specific protein targets in single cells with a confidence that far exceeds what we're able to do today. And in fact, just to add a little bit more urgency to this problem, benchmarking, trying to understand the current state of the technology, you will find that roughly a good estimate is about 50% of the immunoreagents that are available today do not work as expected, right? And I don't know if any of you run a lab here, but certainly if you were a PI, you're hard at work raising funds for your research group. These antibodies are actually quite expensive, and then to know that about half of them fail, which means your assays are going to be essentially garbage, is just a horrible position to be in, right? Let alone the person who's trying to actually implement the assay and interpret the data, right? It's kind of a very big issue. Anybody here ever done flow cytometry? Okay, so classic workhorse single cell measurement tool. Again, it's an immunoassay if you're using an antibody stain, right? I would say that's the premier single cell protein measurement tool. But it still suffers from that issue. So my group, what we've tried to do is think about, are there ways to supplant or completely obviate the need for using an immunoassay? Let's just get around them completely. And we've done that by using different approaches that draw on this microanalytical systems, nanotechnology, and materials as well, light-active materials. And so our work really spans all the way from the bench to the bedside. So in the case of the HER2 example I gave you earlier, it's more clinical. Because if we can make that high specificity measurement, we can say something about treatment, right? What should be taken. But clearly these types of assays, these immunoassays, are also useful at the bench as well. And the key is we want to figure out what proteins are present in a particular sample for a particular question that we're posing. So we've done everything from develop platforms that allow us to screen thousands of different conditions and thousands of different samples, so these screening type platforms. One huge area where there's been a gap is in screening of what we call biospecimen repositories. So you may not know this, but clinical laboratory facilities, clinical medicine facilities over across the country and the world house repositories of biospecimens. So if you have a tumor removed, a biopsy, samples of that tissue are going to be archived as part of these repositories. Also, blood specimens are also taken. I actually work very closely with a uro-oncologist at Stanford Medical who runs a biospecimen repository for prostate cancer. So since 1989, she's been developing this 50,000 sample repository of blood samples and other samples from men who've been diagnosed with prostate cancer and tracking and collecting samples from those men longitudinally, so as a function of time, which is fabulous because we can then tie their outcome to what the development of the disease looked like to try to better inform treatment regimens for men in the future who may be suffering from prostate cancer. In addition to that, there's a couple other areas we work in. I just want to mention these, one of which I won't talk about much today. Certainly here at UIUC, there's some great work happening in this near-patient diagnostics area, something that people often call point-of-care diagnostics as well. And that's an area that we've worked on, mostly looking at what are called proximal or compartment fluids, so tear film fluid, oral fluids, cerebrospinal fluids, synovial fluids as well, all with an eye towards if we can make real-time protein measurements in compartments next to disease, so say synovial fluid with an autoimmune disease, for example, or arthritis, we might be able to tell how that patient is progressing and be able to modulate treatment based on response. Then the last area that I will focus a little bit on today is being able to make these protein level measurements, as in the case of the HER2 positive breast cancer, at the single cell level. And that's something that's been a challenge. So I'm going to start by doing a little bit more of a deep dive to tell you about some of the classic ways that measurement scientists use to overcome this issue of the immunoassay, right? So the immunoassay If you don't have a specific antibody, you're not going to get a specific signal, right? So that means the answer to your question is going to be squishy. You don't know, right? You don't know how much signal is coming from your target and how much signal is coming from something else when you run an assay. So one of the ways that people have worked to overcome this lack of specificity with immunoassays is to do an immunoassay, so to come in and probe a particular sample with an antibody against your target of interest. But before doing that, researchers have developed tools that will fractionate your sample, so divide it into different fractions of sample. And a really powerful way to do that is to, in the case of the single cell, lyse the cell, break it open, and then use something that's called electrophoresis to resolve the proteins or separate them based on differences in size. How many people here have ever done polyacrylamide gel electrophoresis, agarose electrophoresis? Could you raise your hand really high for that one? Because okay, so quite a few people actually have. For those of you who haven't, I will give you just a little bit of an introduction because it's critical that you understand what that is to understand the rest of the talk. So essentially, you have here something that's called a gel a slab gel. And this essentially, we are looking at the top of it. We introduce sample into each one of these little regions. We apply a potential between the top and the bottom of this little slab gel. It's a polymer. And we basically add our sample here, which is a protein lysate, let's say, apply that potential, and proteins migrate through that polymer network. Okay, small species are going to migrate really fast, so they're going to end up being these little bands here. They've raced to the end of the gel, the separation lane. Slow species maybe are going to have a hard time rotating and migrating through the polyacrylamide gel, so they're going to get stuck up here near the top, right? So basically it's called sizing. We're able to back out protein size by electrophoresing those species through this polymer network. After we do that, oftentimes we take those resolved protein bands from each sample and print them, if you will, what we call blot them, onto a hydrophobic membrane. So we're basically moving sample, now fractionated, from this very small pore size polymer into a membrane that has very large pore size and can hold the material on it through hydrophobic interactions. So that's what this little image is here. We've moved protein from the gel into this polymer, this hydrophobic polymer right here. And then the last step that we do is the immunoassay. So we incubate that membrane with the antibody against the target of interest. Our target lights up, if you will. It binds with the antibody, and if we have a fluorophore on our antibody, we can read out that signal and say, yes, not only is it there, because we get a positive signal, we know the size of that species. If you think back a couple of slides, I talked about these truncated isoforms of HER2. You would imagine, if I'm looking at cells that have this P185 protein, I would have this one band here, let's say that's 185 kilodaltons. But I mentioned there's other shorter or truncated isoforms. Those would come in as smaller species down here, right? They would migrate faster through the gel. So if I'm looking for truncated isoforms of HER2, I would not only see one band that corresponds to the 185, I would see smaller bands at the bottom of the gel as well, and that's how I would get my specificity, right? Because of differences in molecular weight. If you think back to the flow cytometry or the ELISAs or the immunohistochemistry, I just get a signal. I have no information about the size. This gives me information about the size. So you can see that this assay is actually really perfect for looking at things like truncated isoforms, right? Because I can get size information and then I can get reactivity with the antibody as well. And so that's why we've been very interested in pursuing study of these truncated isoforms. Couple of issues. I don't know if anyone here has taken a mass transport or heat transport course before. Could you raise your hand if you have? Any sort of fluids also I'll include as well. Okay, so as an engineer who's interested in transport and optimizing systems, I want to point out that there's a couple of really big transport-related issues that center on this assay. I don't want to understate it. This assay is a workhorse assay. Any biological lab that you go into in the US and in the world are going to be running Western blots. Okay, it is a core assay for sure. And I think any of those labs that you go into, if you ask someone who's running the Western blot, how's it going, they're going to bite your head off. Because it's a horrible assay to run. Who here has run a Western blot before? Oh my gosh, okay, so I think I'm probably preaching to the choir, if you will. that assay can take anywhere between 5 hours and 25 hours to complete, right? And then when you get the assay readout, oftentimes it's not even quantitative, right? It's just it looks a little bit brighter band or darker band. And then if you have some sort of failure with your assay, you have to start all over again. And then I think one of the biggest concerns is the assay itself requires a tremendous amount of starting sample mass. There is no way you can do this assay on a single cell. There is no way you can do this assay on a single cell as implemented here. And of course, many of you probably realize that in diseases like cancer, cell-to-cell variability is where it's at, right? That's the information we need to know to help fill in the blanks and how that disease is going to progress or respond to treatment. So these are the issues. The assay can be long duration. We can have this difficulty with moving those sample bands around. We can lose material. Large species may transfer very slowly to this membrane, whereas small species are just going to shoot through that membrane and get lost in the buffer solution around them. And then lastly, we oftentimes have to do something like an overnight incubation to complete the immunoassay. And that's because we're in the diffusion-limited regime, right? We have to make sure that we get to equilibrium for our readout. So my group has really asked, how can we take an assay like this? Again, it's a workhorse assay, but with our understanding of transport, our understanding of the unmet, how can we develop a new class of assays that will allow researchers and clinicians to make these measurements and provide deep profiling of large numbers of samples to be able to answer clinically relevant questions? So there's been some really exciting developments in the area of these so-called multi-stage assays, right? So the Western, there's sizing, and then there's the immunoassay, so it's multiple stages to the assay. It's not just a simple immunoassay. There's some new instrumentation that's on the market. Protein Simple has a really cool system that basically makes a Western blot look something like this. It's all in kind of a box. There's been some work with these really large format gels. So if you run a conventional Western, your gel is probably going to be about that big. Most people, these formats are really big gels. They use a nano printer to print little spots of different samples in the gel and then run electrophoresis. They do transfer material to different membranes and do imaging, so it's still kind of a cumbersome workflow, but really, really large numbers of samples. And then also out of Michigan, Bob Kennedy's group has done some beautiful work on doing separations in capillaries, so to get that first piece of information, and then basically almost using them like a ballpoint pen and dragging them over a membrane so that as the protein bands or peaks come out the end of the capillary, they're printed onto that membrane, that blotting step that I talked to you about. So a lot of these innovations are really promising, certainly, but there is one thing that they all have in common that was not really suitable for our work, and that is, and I'm a mechanical engineer, so it's going to be funny that I'm saying this, every single one of these assays required pretty intense automation and robotic handling, right? And again, I'm a mechanical engineer, so I would say one thing I love is, sure, robotics. It's awesome, right? It can do a lot in terms of throughput and automation. But when you go into most single investigator biology labs or a pathology lab, you don't see robotic instrumentation in those labs, right? The way the Western is run is right here. Here is my bench. This is my setup. I'm going to run my Western right here, right? I don't want to go to a core facility to do this. And so as engineers, as designers, we want to design our technologies for adoption, right, for uptake. We want to disrupt the way measurements are currently being made in a way that will have a big impact. And so for us, our choice was, let's try to develop this system so that we can get single cell measurements with the Western, but that does not require some heavy infrastructure and robotics for handling, right? Plus, I think you can do it more elegant than using robotics in this particular case. And the solution that we've worked around, the framework that we've used to be able to develop a fully integrated multi-stage assay without the use of robotics is the microfluidic formats. And those are very powerful from the perspective of integration, so being able to integrate different stages of the assay. So I'll just briefly use this microfluidic approach, kind of this illustration, to tell you maybe how people have been doing microfluidic multi-stage measurements, and then I'm going to walk you through how we do it, which is quite different. So typically, people will use a microfluidic chip that looks something like this. So you're looking down on that chip right here. You can see these little white lines. Those are microchannels. How many of you have used microfluidic chips or touched them? Even in the last day, it still counts. Could you raise your hand pretty high so I can just see who isn't? Raise their hand. OK, perfect. Essentially, I will say this, and I am a microfluidicist, so I'll be disparaging myself, but they're highly functionalized glass microscope slides is basically what they are, right? So they're pieces of glass that have these microchannel networks etched into them, and then a top plate is sandwiched on, for example. There's other ways to do it. And you have these little enclosed microchannels that can route and manipulate fluids and do separations to make measurements. All of the microchannels I'm going to show you today, well, most of them, are about the diameter of a human hair. So when you think about the microfluidic channels, think about a human hair being that channel, right? And that's what you're looking at here. You have a network of channels that are about the size of a human hair. There's also an intersecting channel right here. And so if we think about this concept, so we're all designers now, we want to develop this multi-stage assay in a planar device, no robotics coming in, although I will mention robotics one point later in the talk. How will we do that? Well, if we used a framework like this, we might imagine using this horizontal channel to do the protein sizing. Right, we want to do electrophoresis to get molecular mass information so we can distinguish those different truncated isoforms from each other. And then what we might do is take each of those bands and shunt them into these intersecting array of channels. And then you might imagine in each one of those channels, I might have an immobilized antibody, let's say, against my target. So I do my immunoassay in those little network of intersecting channels. So I can basically say, does this fraction of protein from the first stage of the assay contain HER2 protein, right? And so if that protein sticks there, then yes, it is HER2 positive. If it goes right through immobilized protein, maybe it's not HER2 positive. No one's actually done that to my knowledge or used this for a Western blot, but you could imagine that. So I myself used an approach like this earlier and also in my own dissertation research to be able to do something that's known as 2D electrophoresis. It's another multi-stage assay, not an immunoassay. And I will be the first to say that this approach, what I call a discretized design strategy, is powerful. It lets you integrate different types of measurements. They require different chemical conditions in each channel. But it suffers from some serious limitations, and that was what we were quite interested in overcoming. And I'm just going to kind of skip, maybe I'll just use this guy right here for today. Basically, our system, the ability to measure multiple proteins in our system is locked in to the geometry of the device. Another way to say that is I can only do as many immunoassays as there are intersecting channels, right? So if I have one assay and I'm looking maybe against 3 targets, I want 3 immunoassays, maybe I only need 3 side channels. If I want to do another assay that requires 30, I'm going to need a completely different chip design, right? So I'm kind of locked into that chip. It's a little bit more complex than that, but basically, that's an issue. And then the second aspect, and Don DeVoe from University of Maryland took this image, is that when we work really hard to get those separated protein peaks that I showed you from electrophoresis, if we start moving those peaks around in the chip, we're going to get band broadening, something that we call dispersion. We're going to smear those peaks out, right? So you have one protein band at a certain molecular weight, you handle it and move it around on the device, it's going to basically spread, owing to diffusion, if not other dispersion processes. So you can get a real mess, basically. You can lose information by handling the sample. And so the way that we decided to come around this was basically to say, let's forget this these discrete intersecting channels. Let's forget them. Let's use instead something that's maybe more continuum, right? So every peak that's separated, it can get sampled into a region of the device that is not discretized by those intersecting channels. Something like a microchamber, okay? So what does this mean? This means if we have a microchamber, so this is about 1 millimeter on a side, so it's much, much smaller than a stamp, a postage stamp, even though you might imagine that it looks something like a postage stamp here. We inject a plug of our protein lysate, let's say, at the top of the separation axis. We apply that potential just like we do with the slab gel, and the proteins migrate from top to bottom through the microchamber. So they size, right? They distinguish themselves by differences in size. Then, after we finish the separation, we might imagine that we could switch the potential 90 degrees and then basically force the species to migrate laterally across the chamber under the action of electrophoresis. So in the case of the Western blot, this would mean we do sizing from top to bottom. We get those truncated isoforms resolved from each other. And then we have, say on this side of the device, antibodies immobilized that allow us to do the immunoassay. So if your protein sticks there, you know it's reactive against the antibody, and you know what size it's at as well. Okay, so in order to do this, and I think as is relevant to this course that you're in right now, we had to develop some new methods to basically photo pattern different types of polymers in this 1 millimeter by 1 millimeter microchamber. Okay, so we developed some photolithography approaches that allow us to get pretty good resolution for different functionalities. So this is what it looks like. Separation, transfer, binding. And that is 57 seconds, right? That is 1 minute. So if you've ever done a conventional Western, if you haven't, I mentioned 5 hours to 25 hours, we're now able to do it in less than a minute, right? And that is the power of, one, using microsystems for integration. I didn't have to do any sort of manual transfer here, right? The chip does it for me. And the power of scaling. So all of the transport processes I talked about a moment ago, this electrophoresis, the binding reactions, they can all be performed in a way that benefits from miniaturization. So it makes those processes more efficient, if you will, than on the macro scale. Okay? So that was our first iteration, I'll say, of the on-chip Western blot. If any of you know anything about engineering design, or probably in your own work, even if you're not an engineer, you know oftentimes the first approach we take we don't end up liking as much as maybe we thought we would, right? We oftentimes iterate in terms of our solutions. And that is an absolutely cornerstone aspect of the engineering design process. We are able to prototype, test, and then come back and analyze and assess what did well and what didn't do well, and then go into another iteration cycle. So what did we like about this? Well, it helped us invent some new fabrication approaches. We were able to do a very fast Western blot, a 1 minute Western blot. What we didn't like about it, however, was the fact that this little microchamber, highlighted here in yellow, is only part of the device, right? So I'm holding this glass chip. We've got the microchamber, the heart of the device, and then these whole channel networks surrounding it. So if I want to start thinking about scaling this up to single or down to single cell level, I don't want to make a measurement on one cell, right? I want to make a measurement on thousands of cells. And if I'm going to do it with this solution, I'm going to need basically a bunch of real estate to be able to complete that, right? It doesn't scale up nicely. It's actually kind of bulky. No robotics though, right? And so with a graduate student in my group a couple of years ago, we challenged ourselves to say, what is the minimum framework that we need to be able to complete a Western blot? And our answer was a separation axis. We need one dimension for sizing, right? We need to be able to resolve the species from each other using electrophoresis. And so as a separation scientist, that means we need two wells, one for the sample and one for the waste, and then an axis here so that we can do sizing. Okay? And so what we ended up coming to in an iteration, coming to develop was basically a microfluidic channel, again, the size of a human hair in diameter. You introduce your sample at the head of that chamber, or that channel, I'm sorry. You apply the potential. You separate species based on size. So the time is right up here. This is a 20 to 200 kilodalton protein ladder, so very wide range. We do a little bit of fancy stuff with the polymer to get a really nice sharp interface here, and we actually use a discontinuous buffer system as well to give us really nice stacking. I won't go into that today, but we're able to get very, very efficient high-resolution separations very quickly on these single channels. There's 3 of them because it's a triplicate measurement just for us to assess the reproducibility. Okay, so after that, fine, we've completed the separation. We now have the different protein species resolved. There's these small truncated isoforms and the large P185 protein. But we have to somehow transition this from a separation into an immunoassay, right? And so the way that we did is using a very, very common photoactive moiety called benzophenone here, coupled with methacrylamide. So we're able to interlink that molecule with our polymer, this polyacrylamide gel. That lets us basically do sizing And then with a brief exposure of that channel to UV light, so a couple of seconds to tens of seconds, we basically abstract hydrogen from any CH bond that's present in our sample, immobilizing protein now on the gel. Okay, so there's no manual blotting step. The proteins are locked in place in the gel using light. Okay, so it's a light-active polymer. A key thing to note about that is a covalent bond that forms. It is not a hydrophobic interaction, as I compared to with the conventional approach. And covalent bonds are much stronger than hydrophobic interactions, so it's a very stable, which helps us do some pretty cool things with the proteins after they're locked in place. But then for the rest of the immunoassay, we basically use a potential to drive in antibody and then secondary antibody often to get a nice readout signal, and then you can start to see the peaks where the antibody is retained, right? So it's a Western in a single channel. And this we've done optimization of the transport and reaction kinetics to be able to get the assay to be high performance, so detect very, very small amounts of protein. And I won't talk about our engineering work on that today. So we've extended this assay. We've been able to look at wide molecular weight ranges, so here from 6.5 kilodaltons to 116, which is important for some questions that we've been posing as related to prostate cancer. We've been able to look at raw cell lysates. We're able to come in and probe that channel with multiple antibodies, something that's called reblotting, so you can get more information out about what targets are present without having to do another separation step, which is very nice, especially if you're working with a sample from a biorepository where you can't go back in and get more sample out because you've used it and it was from a patient that was collected in 1990, let's say. So that's essential as well. And then also, the last thing I'll note about this microfluidic format is we've been developing the Western using the microfluidic format for point-of-care diagnostic as confirmatory, not a screening diagnostic for HIV infection. I don't know if you know HIV. There's typically a screening assay that uses an oral fluid. If it's an okay assay, it has a lot of false positives. If you get a positive back from that oral fluid assay, you have blood drawn and it's run on a Western, which is a high specificity assay, right? Here in the US, that's not a problem because we have clinical laboratory facilities where those Westerns can be run day in and day out. If you are in a resource limited setting, however, and you have that positive result come back, it's actually pretty hard, can be pretty hard for that person to get back for a blood draw and then the clinical laboratory test to be run. So what we're trying to do is think about ways that we can take this point of care format but make it confirmatory quality. So there's just one step for diagnosis as opposed to this multi-step process. And then what I mentioned before, the major limitation of the first approach that we used is this concept of scale up. If I wanted to do, say, 100 of these Westerns at a time, it was hard to scale up from that chip design I showed you earlier. Whereas here, this is a glass microscope slide, and you can see we have 48 micro Westerns all packed into that slide, right? So we can run them all at one time. I will mention students who, these glass micro devices, they're actually quite expensive. They're difficult to make. We can reuse them. We've developed protocols to do that. But I think a couple of my grad students were just sick of spending time in the clean room, if you can believe it. And they decided, you know what, let's forget the channels in these glass devices altogether, and let's just take polymers and pattern them on flexible materials. So instead of creating the polyacrylamide gels inside the microchannels, the size of the human hair, let's just pattern them on top. So there's no walls at all, right? And so what you're looking at here is polyacrylamide gel, so that separation matrix, but we've used light to be able to photo pattern little injector wells and different geometries on these devices, which has allowed us to do high-scale screening of conformation changes in a different assay that I won't talk about today. Okay, so that's kind of the story about how, as engineers and separation scientists, we've developed these tools, tried to understand really what the needs are, and then gone through an iterative process to get to the point where we're able to get the performance that we want. I have a couple minutes left. I want to complete the story, if you will. I have been motivating this question by saying these immunoassays are limiting, they have low specificity, and we cannot make these measurements on single cells. Right? So we've taken this assay and developed it for bench top measurements in biology labs and then these diagnostic devices, but have also now taken this assay and developed it for single cell measurements. The way that we do that is very similar to what I showed you before, but I'm going to highlight a couple of differences. Instead of the micro device I just showed you that has those microchannels packed on a microscope slide, we essentially use a microscope slide as a substrate, so you're kind of side on here, a little bit off axis. And then we have this polymer that, again, we're patterning just free and open to the air. So it's a thin polymer layer that's patterned on top of the microscope slide. It's about 30 microns thick, excuse me. And in the polymer layer, we've stippled microwells. So each of those microwells is about 30 microns in diameter. Human hair is about 100. And so essentially what we do is we take this geometry. This is our artist's rendition of the geometry. You have that polymer with the microwells. We use gravity to settle cells into the microwells. So basically 1 cell per well. So cells in the wells right here. We basically come in and chemically lyse the cell, so you can see the debris of the cell left in the well. Apply a potential across the device to do electrophoresis for protein sizing. And then come in with that UV photoactivation of the benzophenone that I mentioned before to lock those protein bands into the gel, and then probe the immunoassay steps right in the gel. Okay? And so using an approach like this, and this is a confocal image of a neural stem cell in one of the microwells that's created in that polymer matrix that we use, we're able to make single cell protein measurements using this Western blotting device, right? And I think a key aspect that I'll mention here is the only reason this is possible to be able to make protein measurements in single cells on a lysed cell like this, the only way this is possible is by using the physics of the system. And basically for us, that physics means we need to lyse the cell and do every single stage of the assay as fast as possible so that cell does not, the lysate does not get diluted, right? Because once that lysate gets diluted, my sensitivity is too low. I'm not able to detect the proteins. For those of you who have done flow cytometry, that's kind of the far extreme of this principle because you don't lyse the cell at all, right? You keep that cell intact. There's very little dilution of the components in the cell. In our case, we do lyse it, but we do everything super, super fast so that we outrun diffusion, basically, is what we do. And that's kind of some detail on that. So what does this look like? This is a bright field image with just a little bit of fancy lighting that shows you that microscope slide with these blocks of microwells. When we complete the Western blot, one of those blocks looks something like this. So we had cells per well. You can see those cells settled into the microwells. We lyse them, we do electrophoresis, lock them into place, and then probe with the antibody here against beta tubulin and GFP. So you have 2 peaks that you can measure, and that's the Western on the single cells. I won't talk about it today, but we've looked at several different questions. Everything from differentiation, in this case of neural stem cells, to try to understand how these species develop and the variability in differentiation among a population. One thing I will mention for today, which is really the crux of why anyone does a Western as opposed to just flow cytometry, and that's really found here in this example. At day 0, we had a single neural stem cell. We lysed that cell and did the electrophoresis to do the Western. Intriguingly, this particular marker of neural stem cell differentiation, so a lineage is nestin, is the name of the protein. When we look closely at the results for the nestin protein, we found, and here's the distance, and this is the fluorescent signal. We found there were 2 peaks, so they're both green here, that are associated with nestin. It turns out there's what looks to be an unexpected isoform or complex of nestin with another protein that migrates at a different molecular weight than we would expect. So if I did flow cytometry, I would never see that. I would see, yes, nestin is present, but I would have no idea that there were 2 different sizes of nestin that were expressed in this cell. So that's just an example of an unexpected result that you would not see with another approach. Like flow cytometry, we can watch these signaling cascades as a function of time. So here on the x-axis, we're looking at phosphorylation of MEK, which is a particular MAP kinase protein. On the y-axis, phosphorylation of ERK, which is another MAP kinase. And you basically see this phosphorylation event, and then it propagates up to the ERK, and then over time it kind of collapses back down to the initial state with some rare cell outliers that maintain high levels of phosphorylation. A couple things that I do want to highlight. So again, one of the key design characteristics of a device like this is the ability to keep that cell material as concentrated as possible. So after we lyse the cell, we don't want the material to diffuse away. Couple of issues that we came across. You're watching the assay here. The cells are lysed. You're looking down. Those are the microwells. Here's electrophoresis. It's going to go dark because we do this UV exposure. And then this is the material that's retained inside the cell. This is a fluorescent protein, so you can watch the process. Major losses of material. So during lysis, we lost almost half of the material. And that's because diffusion is so fast on these tiny length scales, right? And then through the top of the gel, as those peaks migrate down the channel, we actually lost almost 20% of the material, even though those separations require just 30 seconds to complete. So you're seeing we're losing almost 60% of the protein mass. If you do numerical simulations or just take a look at scaling, you basically see the way that we were introducing the lysis buffer. We had our little chip and our microwells, and you just pour the lysis buffer over the top, and then the cells pop. They lyse inside the microwells. That convection of solution over the top of the microwells basically just dips into the microwells and pulls material out, right? So it kind of entrains the material and pulls it out of the microwell. So we've developed some systems that don't use bulk flow or convection at all, right? Because that was significant losses. In addition to that, we've been able to develop the assay to look at multiple proteins, so different antibodies against different targets in the same single cell. Here up to about 14 different targets in that same cell. And then I want to show, and I won't go into this much today, but with the multiplexing, so this is one cell that we've looked for all of these different targets in, and these are the results for each of those targets presented here on a biaxial plot. We're able to do quantitation of how much protein is present, which means that we can come in then and start doing correlations and see which proteins are correlated in expression level and state in specific systems. And this is some questions that we have related, again, to that HER2-positive breast cancer. And then lastly, I think I'll show you just maybe 3 very brief more things, 1 or 2 slides each. In addition to being able to do these single cell level measurements, as I mentioned with the HER2 protein, you can often expect that protein to be present in different regions of the cell, right? So I mentioned the membrane, the cytoplasm, and the nucleus, right? And so we're interested in figuring out, could we use a method like this to selectively Western blot the cytosol, say, versus the nucleus of the cell for a particular protein of interest. And so I have a fantastic, almost 4th year, I guess he'll be starting his 4th year, graduate student who figured out a way to basically seed the cells in the wells, lyse just the membrane, leaving the nucleus intact, so the nucleus just sits in the bottom of that well. We apply the potential, we take all that cytosolic protein and electrophoresis it away from the well, lock it into place with a UV light, come back, lyse the nucleus, right, that was sitting there in that microwell, and then just flip the polarity of the applied potential to drive the protein from the nucleus into this other direction. So we can get cellular compartment-specific Western blotting, right, which is pretty amazing to me because these features are really, really tiny, right? So the cells can be anywhere from 10 to 30 to 50 microns in diameter, with the nucleus being smaller than that. And so those are some of his preliminary results. So that's one aspect. We've also taken this tool and coupled it with conventional instrumentation. This is coupling with flow cytometry. So basically being able to do flow cytometry, and this is kind of what typical readouts, each of those dots is a single cell measurement from flow cytometry. You can select or gate on a particular population and then basically spot just those cells from the nozzle of the fluorescence activated cell sorter onto our micro device. And then it looks something like this. And then basically come in and do Westerns just on those selected cells, which allows you to get membrane protein information and then do the Westerns on the different compartments. This is some work that we've been doing, and I won't go into it much today, but in modulating the materials that are used for this device. So the device I showed you, you look down on it, it was a layer of polymer with the microwells in it. We've been able to tune the porosity of that polymer, and what you're looking at here is for each microwell, aligned with it is a tiny gradient gel. So a little separation region that has a very large to very small pore sized polymer, which is what's typically used to get high resolution separations for electrophoresis. It's very effective at what we call molecular sieving to resolve different protein targets. And then the last thing I'll close with is to kind of complete the story. I mentioned earlier that it's difficult and in some cases impossible to measure these truncated isoforms with single cell resolution. What I'm showing you here is a HER2 positive breast cancer cell this is the western. Here's the top of the channel, so this, or the top of the lane, so this is where your large protein would be, the full length protein, this guy over here. And you can see for a HER2 positive cell that we see that, but you also see a much smaller protein that's migrated from that cell. That is the P110 truncated isoform. So we've been able to resolve this full length from truncated isoform in a single primary breast cancer cell. In this case, that's an engineered cell line, but we've also been working on these biopsy samples as well. And that's where we wanted to get with being able to help differentially diagnose breast cancer for treatment decisions. So with that, I clearly have to thank my research group, including alumni who are not pictured here, who have contributed this work that I showed you today. I also need to thank our collaborators, as well as the patients that have donated their tissue samples to our work. I want to thank our funding sources clearly, and it's something that's relevant to you who are in grad school or thinking about going to grad school. Federal fellowships have played a huge role, in international fellowships, in the ability of my students and trainees to conduct their research, and so I definitely have to acknowledge those as well. And I also want to mention I am involved in commercialization efforts for the single cell analysis technology as well. Thank you so much for your time. Appreciate it.
Thank you so much. It was a really fantastic talk. So we have 15 minutes available for questions. So would anybody like to start off the conversation?
I have a
Oh, cool. I was going to say it's hard to go after lunch, but I guess.
in a biopsy, in an actual sample
Sure.
from another person or a patient.
Yep.
How do you measure, like in a single cell, how do you measure the different proteins? And how do you know that from one cell to the next is the same proteins are present or expressed within those cells?
Right. So the question is from a biopsy. If you have a biopsy of cells, so a mass of cells that are collected from a patient, how do you measure between the different cells the protein expression levels?
And if they're the same.
And if they're the same.
Like, how do you know that?
How do you compare them,
Yes.
basically? Okay, I'll answer it, and then you tell me if I am answering or not. Okay. So essentially what we do is we take the biopsy or the tumor and we dissociate, so we chemically dissociate the extracellular matrix and kind of break up the tissue so it's a suspension of cells. And then once those cell clumps are kind of broken up, we settle them into the microwells. We typically use a housekeeping protein, so something that we know is correlated, for example, with cell size, beta-actin or GAPDH. And we probe for that in every cell so that we can then normalize the protein signal of our target to that housekeeping protein and then do comparisons between the cells. So for us, when we want to do relative comparisons, there has to be some kind of handshake, and that's that housekeeping protein that we're comparing. Now, it's a tricky issue, and maybe this is why you're raising this. Their GAPDH, for example, is a housekeeping protein that's been very widely accepted to be able to normalize signal. Well, it turns out that GAPDH seems to have a lot of heterogeneity in cancer. So if you're using that marker, you're kind of confounding two signals. And so for us, we've chosen to use several housekeeping proteins, not just one. We also do imaging beforehand so we can get an estimate of the size of the cell, which can also help us to normalize to cell volume as well. But I would argue that it is not a clear-cut answer with proteins in particular. Yeah, it's a great question. Yeah, excellent. Maybe here first.
So in typical Western blots, there's this blocking step where you have to block the
Yes,
sites.
yes.
How do you address that issue with your microfluidic platform?
Yeah, great question, great observation. So the question is, in Western blotting, so after we do the separation and we do blotting, that physical blotting to the membrane, that's a hydrophobic interaction. So we're transferring that protein band onto that membrane. You already know this, but onto that membrane. When I come in with an antibody, it's going to stick everywhere as well, right? But I only want it to stick to the target of interest, so I have to block, we call it, the rest of the, chemically block the rest of the membrane. And we typically use something like a sticky protein, BSA or casein or something that's going to just basically occupy all those hydrophobic locations, just allowing our protein band to be there and interact with the antibody. Well, it turns out that we've been using, we don't use hydrophobic interactions, right? We use this benzophenone moiety that covalently binds to the proteins. That moiety is only reactive when UV light is shined on it. So I think the timescales for the kinetics of that reaction are sub-microsecond kinetics. So basically, as soon as I turn off the UV light, that polyacrylamide gel is now inert. It's basically inherently blocked. So the only reactive sites are where the proteins are. And that's one reason that we often, as separation scientists, use polyacrylamide gel, because it has very little non-specific adsorption to it at all. So it's we turn on and off the activity of the gel with the light. Yes, yeah, absolutely.
Thank you, Professor Herr.
Sure.
I want to ask, in the single cell like Western blot sheet, like a lot of acids, it's like, can you compare like how much a certain type of acid forms a cell express, or is like on and off? You just know if it's expressed or not.
Great question. Yeah. So can you be? I'm just rephrasing the question. So is it just presence or absence, or can you get some information about the level? Yeah, quantitative. Absolutely. So we have used this assay to perform quantitative protein level measurements, and we also use qualitative as well across the spectrum. So we can say high to low, medium as well, or somewhere in between. But when we do careful control of losses at each step, we can then back out quantitatively how much mass do we think is there. And it's a kind of two different assay designs, because for quantitation we have to be a little bit more rigorous, which takes a little bit longer. But I skipped this one slide. We've done quite a bit of work to try to understand how quantitative can we make the assay and what does that take. So this is an example of, this is the number of target molecules. So here we're using GFP. We use that because we can actually measure fluorescently how much protein is there, right? And then on the y-axis, this is how much antibody, or the signal-to-noise ratio of the antibody. So we can come in and generate calibration curves, basically, that let us correlate the SNR of the antibody probe with how much protein is present. So you can generate these calibration curves as well, if you're interested in that, and in some cases we absolutely are. It's a great question. Yes, absolutely.
Based on single cell assay, if we think about what is the biological significance of this, and also when we think about treatment and clinical studies, we don't even discuss about tissue or organ, we discuss about the whole human body.
Yes, yes.
So also when we take some drugs for the treatment of certain like diseases, cancer, and if we want to see that it is going to affect at some particular protein or genes which is overexpressed in particular cancer like HER2 or breast cancer.
Yeah.
So I'm thinking that when we take some drugs to treat that particular disease, and we test that this drug is responsible for down-regulating of the gene and down-regulating that particular oncoprotein, then some cells, if we are working on single cell assay,
Yes.
some cells become resistant to the
Right.
drug.
That's
So if we go through that assay, probably we would not take the same, the cell we are taking would not have the drug absorbed inside that cell.
Right,
So
right.
in that case, how would that
Yeah.
assay be clinically significant?
Yes, that's a great question. That is a great question. So if I could just summarize, maybe, and I'm sure I'll miss some important parts, but I apologize for that. So the question is, to paraphrase, that an individual who may have cancer, as an example, through the course of therapy, there are these proteins maybe that are overexpressed. So HER2 is an example that I gave. So the cell surface is just decorated with these extracellular domains. In the course of treatment, you may get some response, right? You may kill off some of the HER2 overexpressing cells, but maybe some of those cells that don't kill off will become resistant to the drug, right? They'll evolve essentially to be resistant to the drug. So the question is, how could you use a tool like this to monitor and inform treatment if you're basically destroying your cells when you're doing your assay, right? And so I think that's the question. So one way to do that is to figure out how you can take biologically different measurements as a function of time for that individual. The one way that we've been working at that so far with breast cancer, because the biopsies are somewhat invasive. I mean, it's possible to do them, but typically the whole tumor is removed, right? We don't leave the tumor in for breast cancer. The way that we've been working to do that is actually collaborating with Stephanie Jeffrey at surgery at Stanford on looking at these circulating tumor cells. So is there a way to get what people are calling a liquid biopsy? So basically take a blood sample and look for these circulating tumor cells in there. There's a whole bunch of debate about those cells, how important or unimportant they are. I mean, that's the topic of an entire conference, I would say, and I'm sure there's been many. But in our case, we've been collecting these circulating tumor cells from patients who have gone metastatic after treatment to try to figure out, are there any correlations with any of these key pathways and the progression of their disease. So to be clinically relevant, here, I think upstream, in this example, you would be able to inform therapy selection using the resected tumor. Yes, we have these truncated isoforms. We need to go with the tyrosine kinase inhibitors. But during treatment, as your question is, you need some way to monitor the response of the individual. And what we've been looking at is working with the circulating tumor cell specialist to try to figure out if that gives information that can guide therapy. Does that answer your?
And also, when we do the conventional Western blotting,
Yes.
we take almost 100,000 cells.
Yes.
Or 500,000 cells or 1 million cells
Right.
to get very little amount of protein.
Yes.
And when we go to the single cells, do you think there would be too much protein in a single cell to monitor this protein expression?
Yes. So the question is, with the conventional Westerns, so the Westerns I showed you earlier, if you haven't done them before, we typically use 10 to the 5 or 10 to the 6 cells in one sample to be able to make the measurement. Clearly the issue there is we're pooling them. So cell-to-cell variation, we're just basically mixing it all together. We're never going to know, right? In some of those cases, it's hard to make detection even of some of the targets, even after pooling the cells. In the estimates that we've done, and some of this speaks to that, we are able to get down to, in a single cell, 30,000 molecules of protein. Okay, so if there's 30,000 copies of protein, we can use conventional fluorescence with this microfluidic device to detect that protein target. We're working on some amplified readouts now that I think would push us even lower in terms of the number of proteins, pretty easily actually. The important thing to note is transcription factors, many of which we're quite interested in with response to treatment in cancer, are at about the 50 times 10 to the 3 level, and that's right in the range of where we can detect right now. Okay. Further, we've done some analysis of the mammalian proteome, and our numbers, and there's a little bit of debate in the literature about the dynamic range of expression in that proteome, our numbers show that with the assay that I'm showing you here, we should be able to measure about 80% of the proteins in the mammalian proteome. So the lowest expressing 20% of proteins we cannot measure with this approach yet, but the top 80% we can. And in the case of these truncated isoforms, they actually express fairly highly. The main issue is not every cell expresses them. So making sure that you capture those rare cell events is another part of that sensitivity challenge that you're pointing out. And so we're working on some fluidic methods that allow us to, with low losses, route the cells to the microwells and capture them all. So make sure to see those rare events. So really great questions. Thank you.
Do you know what currently limits your limit of detection?
The limit of detection right now is it's really limited just by the mass of material that's there and the readout technology that we're using, right? Certainly people can do single molecule detection. The question is, would you want to do that in a clinical facility? Probably not, right? And so for us, that's a matter of, instead of using just fluorescence readout, there's a lot of chemistries that can actually amplify the readouts and give you much lower sensitivity, which is better, right? And so some of those approaches are compatible with our technology, and are promising in terms of extending it. Yeah.
So the
Sure.
question here, so regarding the time, how long does it take for detection?
Yes, for the workflow. How long does the assay? I apologize, I omitted that. So this particular single cell assay, the workflow that we're using is about 4 hours. And most of that is actually incubation with antibody. So that antibody incubation step for the immunoassay to make sure that you reach equilibrium. So we have some Just submitted a manuscript now, looking at decross-linking the gel in order to get the antibody in more effectively so it doesn't partition out into the fluid phase. Yep.
And what stage in commercialization is this now?
Good question. Yeah. So an alumna from my group and a couple of other engineers and scientists, I think there's about 7 employees now. So they've been working for almost a year and a half to do commercialization. They have 5 pilot instruments that they're deploying to labs in the San Francisco Bay Area. I think that's so when they break down, they can drive over and fix them. And they've secured, I think it's about 1.8 million in angel funding. So they've gotten some good traction there, but still uphill battle in terms of commercializing life sciences tools. That is a very difficult market if you're interested in starting your own company. So things are going well.
Fantastic.
So, follow-up question to that.
Yes.
So it would have to go through the FDA then?
This is, so, yeah, I should make that distinction. That's a very good question. Should this go through the FDA? So the commercialization effort that this company is leading is really focused on life sciences tools and not on diagnostics. And they chose that specifically so they would not have to go through FDA. That's right, that's right. The work that we're doing in the research group, a lot of it is actually focused on diagnostics, because I think there are big challenges there. And quite honestly, I'm not worried about FDA at this stage. We're way too early. So I'll hand that off to someone else.